Hi, thank you, everyone. So as Harinder said, my name is Barb Jusiak, and I'm in physiology and biophysics, and I work in MedSci B building. And feel free to email me if you have any questions. Can everybody hear me okay? Okay, wave if you can hear me. Everybody good? Okay, excellent. All right, so as Harinder said, we'll be talking about innate immunity today. And here are the keyboard doesn't seem to be working. Okay, it's got a bit of a delay. Thank you. All right, so here are the learning objectives for today. Why is the innate immune system important? Who are the cells of the innate immune system? And how do these cells get to the site of pathogen entry or invasion? How do the cells recognize pathogens? How do they kill pathogens? And what other lines of defense do we have against pathogens? So the first question, why is the innate immune system important? And the reason it's important, it's that it's the first line of defense for when a barrier is breached. So what kind of barriers does our body have that separates the inside of the body from all the pathogens outside? Anybody? Sorry? Yes, very good. Skin's a major one. Anything else? Mucus, yes. Very good. So what other body, other than the skin, what other body surfaces are exposed to pathogens? Eyes, yes, very good. What about when we eat? Yeah, exactly. The gut, the gastrointestinal system, whatever you eat is technically on the outside of your body until it gets absorbed. And lastly, how do we breathe? Lungs, exactly. Okay. So we've got all these barriers, the skin, the gut, the lungs, and the eyes, nose, and oral cavity. And each of these has three types of barrier: mechanical, chemical, and microbiological. So in terms of mechanical barrier, first of all these surfaces include epithelial cells joined together by tight junctions. So there's a physical barrier for the microbes trying to get in. Secondly, there's movement of air or fluid across your skin and in your gut. And then in your lungs, you have continuously beating cilia that move the mucus up and out of the body. And in the eyes, you also have tears. Chemicals, you have different things like antimicrobial peptides, fatty acids, and in the stomach, of course, you have extremely low pH that breaks down a lot of invading microbes. And lastly, microbiological, you have your normal microbiome that constantly colonizes your skin and your gut, and that outcompetes many of the pathogenic microorganisms. So we have all these barriers, and then the innate immunity comes online once one of these barriers is breached. So here we see, first of all, healthy skin is not inflamed here in the leftmost panel. And then as soon as you have a breach, so let's say you get a cut and some bacteria get in, this attracts innate immune cells, which start secreting cytokines. Also, a blood clot forms to prevent more pathogens from coming in. The cytokines have many functions, but one of them is to attract more innate immune cells coming in, which you can see in this next panel here. Cells start to come in. And also the cytokines loosen the junctions between the epithelial cells and cause fluid to come in. This is called vasodilation and increased vascular permeability. The blood vessels get wider and the junctions get looser. And what happens is, as a result, the infected tissue becomes inflamed, which involves redness, heat, pain, and swelling, as the innate immune system fights against the infection. And here is a table of a bunch of different pathogens that the innate immune system fights and the ways that they get into the body. So the main takeaway from this is there is a lot of different pathogens out there. So you can classify them by how they enter the body. You've got the mouth and respiratory tract, the intestinal tract, reproductive tract, and also opportunistic infections by microbes that are normally found on the body, but typically don't become pathogenic unless the body is in some way weakened. And then you also get pathogens that come in through wounds, abrasions, and insect bites. Okay. So the innate immunity, as we established, is the first line of defense. And here we see sort of a time course of what an infection might look like. Microorganisms enter, infection becomes established and reaches a threshold. When you reach a certain threshold, if the innate immunity doesn't succeed in clearing the infection, then the adaptive immune response kicks in. So the innate immunity is the first quick line of defense. The adaptive immunity comes online later if the innate immunity cannot handle the infection. Then you have the inductive phase, the effector phase, when the adaptive immune response hopefully resolves the infection. And then you have the memory phase, much more about that in the adaptive immune part. Okay. So this is the number of microorganisms over a time course. Here, normal humans, you get an infection, microorganism numbers start to rise sharply, they peak and they decline as they are resolved by the adaptive immune system. If you lack the adaptive immunity only, at first the time course looks pretty much the same, but then the infection doesn't resolve. But if you lack the innate immunity, then you start really skyrocketing with the number of microorganisms, and basically you don't survive because the adaptive immunity never gets a chance to start to come into play. The bottom line is no innate immune system equals no survival. And again, the innate immune system clears infections or holds them at bay until the adaptive system comes online. Here again is sort of a figure that illustrates that. Pathogens adhere to the epithelium, the skin, let's say. Then you get a breach in the barrier, local infection, pathogens come in. There's a local infection in the tissue that involves the activation of macrophages and dendritic cells migrating to lymph nodes to initiate the adaptive immunity. And then later, the adaptive immunity comes online, and you get the infection cleared by specific antibodies and cytotoxic T cells, which are part of the adaptive immune system. So again, the contrast, innate immunity, rapid response time, they have a fixed repertoire and a limited specificity, versus the adaptive immunity, which has a slow response, takes days to kick in, and it's variable and highly selective, and it improves during the course of the infection response, and also establishes memory of specific antigens that it encounters. And these two arms work together to destroy the invading pathogen. Okay. So innate immune system gains access to the site of pathogen entry, recognizes the pathogen based on broad classes of receptors, kills the pathogens, and alerts the adaptive immune system. Okay, so who are the cells of the innate immune system? You start out with a hematopoietic stem cell, which is the stem cell for the entire hematopoietic system. These can then become either the common lymphoid precursor or the common myeloid precursor. The common lymphoid precursor gives rise to the B cells, T cells, and NK cells. And the innate immunity that we're talking about today arises from the common myeloid precursor. So the common myeloid precursor, again, can differentiate into two different things. It can become a megakaryocyte erythroid progenitor. This gives rise to megakaryocytes, which then bleb off into platelets, which function in blood clotting, or erythroblasts, which further differentiate into erythrocytes, the red blood cells. And then the immune progenitors of the common myeloid precursor are the granulocyte macrophage progenitor. So this is the part that we're interested in for the purposes of today's lecture. The granulocyte macrophage progenitor gives rise to the different innate immune cells. So you've got a macrophage and dendritic cell precursor. This gives rise to monocytes, which further differentiate into macrophages, and also to dendritic cells. And then also the granulocyte macrophage progenitor gives rise to neutrophils, eosinophils, basophils, and mast cells. Okay. So let's talk a little bit about each of these. First of all, you've got the neutrophils. Neutrophils are the most abundant type of white blood cell. There's 10 to the 11th of these produced daily, and these are the first responders to damaged or infected tissues. Normally, they're not found in healthy tissues, they're found in the bloodstream, and they are quickly recruited to tissues in response to injury and infection. These are phagocytic, and after phagocytosing the pathogens, they quickly die, and dying and dead neutrophils accumulate and become a major component of pus. So next time you get a cut and it becomes a little infected and you start getting pus, think to yourself, that's a bunch of neutrophils. And here I've got this little video. So let me show this. Okay, there we go. Okay. I'm trying to show you a video of this neutrophil. Okay, here we go. So as you can see, the neutrophil is attracted to the bacteria, which are trying to run away from it here, and it crawls around and tries to eat them. There you go. I find this always a fun thing to watch. So, yeah, and eventually see it engulfs it and phagocytoses it and destroys it. All right. So insufficient numbers of neutrophils result in neutropenia. That's when you have fewer than 1,500 neutrophils per microliter. And there's distinct mechanisms that can cause that. It can be due to decreased bone marrow output, such as due to infection or a congenital defect or a nutritional deficiency. It can be redistribution from circulating neutrophils to the spleen or vascular endothelium, or it can be due to immune destruction, such as autoimmunity. And in contrast, neutrophilia is where there's too many macrophages, defined as over 7,700 per microliter, and that's due to increased production of neutrophils in the bone marrow. So that may be due to a neoplasm, it may be due to infection, it may be due to medications, or it may be due to stress. Eosinophils are a much more rare form of white blood cells. They comprise about 1 to 3% of white blood cells, and they're critical for defense against helminths or parasitic worms and bacteria. They reside mainly in tissues, and they're phagocytic, and they also secrete inflammatory cytokines. So eosinophilia is an excess of eosinophils defined as over 500 cells per microliter, and can be caused by neoplastic disease, rheumatological disease, parasitic worms, allergy, or drug reactions. And here's a picture of an eosinophil. Okay. Basophils are very rare. They're only approximately 0.5 to 1% of all leukocytes, and they are important for fighting parasitic infection. They're the largest of the granulocyte cell types, and they secrete histamine and serotonin. Okay. And then here we have the mast cells, which are also important for defense against parasites, and they are found in the connective tissue and mucosal. They secrete inflammatory cytokines and mediators of allergy. So when you have allergy symptoms, it's the mast cells that are a major culprit, and the mast cell disorders are rare. And lastly, we have the very interesting cells called monocytes and macrophages. Monocytes give rise to macrophages, and the two of them are sort of, as this recent review described them as, they're sort of the tortoise and the hare. The monocytes are the hare. They are short-lived. They're rapidly recruited to the site of infection. They travel in the bloodstream and are recruited out of the blood to infection sites. And they can further differentiate into macrophages. Macrophages are the tortoise. So they come from two sources. There are tissue-resident macrophages, and there are also macrophages that differentiate out of monocytes. And their functions are defined by the tissue in which they reside. So different tissues have their own macrophage populations with specific functions. And they have a whole plethora of functions, including remodeling tissue after injury, and also a contribution in cancer that we're not going to talk about in this course. Okay. And then finally, we have the dendritic cells, which are very important because they are the go-between the innate and the adaptive immune system. Dendritic cells reside in tissues and lymphoid organs. There are conventional and plasmacytoid dendritic cells. And for the conventional dendritic cells, their primary function is to present antigens to T cells, which are part of the adaptive immune system. So the conventional dendritic cells pick up antigens at the site of infection, and then they travel from that site to a draining lymph node for the given tissue. And they present the antigen to T cells, thereby activating them and kicking the adaptive immune system into gear. And the PDCs specialize in viral detection and destruction, and they secrete interferon. Okay. So that's sort of a whirlwind tour of the types of innate immune cells. Does anybody have any questions? Okay. All right. So how do innate cells gain access to sites of infection? Obviously, when an infection happens, as shown here in this lovely picture of somebody who apparently stepped on a nail, the innate immune system needs to reach the site of infection to exert their effector function. There are already, as we discussed, there are already innate immune cells in every tissue, but when you have an acute infection, you need to recruit reinforcements. Okay. So the three major mechanisms are chemokines, adhesion molecules, and inflammatory cytokines. And this regulation is very important because the point is, under normal conditions, we do not want excessive numbers of myeloid cells entering tissue. That leads to problems. So chemokines are signaling molecules that are sort of the traffic directors of the immune system. They tell different types of immune cells where to go. So for example, as shown in this tissue, different organs secrete different chemokines, which then attract specialized types of cells. Okay. And this figure is meant to convey the diversity of chemokines and chemokine receptors. You can see that we have a lot of them in our genome. Every box here represents a receptor, and every oval next to it represents a secreted ligand that binds to that receptor. The naming convention is CXC or CC, and that has to do with the amino acids at the N-terminus of the cytokine, of the chemokine. So CXC means cysteine, any amino acid cysteine, and CC just means cysteine. You don't need to know this, but that's just an explanation for why. And the color coding here shows the chromosomal locus that encodes these chemokines. So the same color means the same chromosomal region. So notice that there are a couple of regions that each encode a whole bunch of different chemokines. And also notice that each receptor tends to bind chemokines that are encoded in the same chromosomal region. And further, the chemokines can be divided into homeostatic and inflammatory. So the homeostatic ones are the ones that are produced constantly. They have important roles in development, and they are confined to specific tissue types. In contrast, the inflammatory cytokines are ones that are induced upon activation, and they aid in the development of the immune response. So they are the ones that become activated when, say, there's a pathogen entering the body, and you need to recruit cells to fight the pathogen. So chemokines that attract innate cells into tissues, for example, CXCL8 attracts neutrophils, CCL3 attracts macrophages, and CCL11 attracts eosinophils and basophils. And so that's the chemokines. And then another way in which we attract cells into specific tissues is through specific adhesion molecules. So innate immune cells express molecules called addressins and integrins. And activated endothelium cells express molecules called selectins and ICAMs. ICAM is an intercellular adhesion molecule. And the addressins bind to selectins, whereas the integrins bind to ICAMs. And this is how you can attract innate immune cells to a specific activated epithelium. All right. Because again, as we showed in this figure earlier, you start out in a healthy state, then there's a breach, bacteria come in, you activate the immune response, and now you need more innate immune cells to stick to the vascular endothelium and to crawl through the vascular endothelium and to go into the infected tissue specifically at this site, not at any other. And this is achieved in part through this interaction between cell surface molecules on the innate immune cells and on the endothelium. And one of the things that activates the endothelium is TNF-alpha, tumor necrosis factor alpha. This is an important factor to remember because it comes up again and again in the function of the immune system. This is sort of a master regulator that activates a bunch of inflammatory responses. So this is a secreted molecule that plays a role in activating the endothelium. And again, one reason this is so important is that we do not want excess immune cells moving into a tissue unless we want that tissue to become inflamed because we're actively fighting infection. Okay. So here's sort of a multi-step process of how a leukocyte can crawl through the vascular endothelium. In the first step, so this is a neutrophil flowing along in the bloodstream, and it needs to move across the endothelium and into the infected tissue. So this neutrophil expresses sialyl residues on its surface. There's a type of glycoprotein that has sialyl residues, and these sialyl residues have specific binding to this protein called E-selectin that's on the basement membrane. But this is weak binding, so it's not enough to make the cell stick completely to the endothelium. So instead, what happens is what's called rolling binding. The cell binds briefly, then it quickly detaches and rolls over and binds to the next molecule and so on. But what it does is it slows it down. And when it slows it down, that gives it the chance to bind to ICAM1 on the induced epithelial cells. And the ICAM1 binding is much tighter compared with the E-selectin interaction. So now with this tight binding to ICAM1, the cell is able to crawl through the endothelium, crawl in between the cells of the endothelium, and enter the tissue, where in turn it migrates in the direction of chemokines secreted by the immune cells already present there. So it's a multi-step process. You have to activate the epithelium to express the correct molecules. You've got the selectins and the chemokine receptor interactions. The ICAMs interact with the integrins. You get the extravasation, which is the process of the cell leaving the bloodstream and going into the tissue. And then once it's out of the bloodstream, the cell moves through the tissue following the chemokine gradient. So it's a complex process. And, sorry? Somebody, yes. Oh, sorry about that. Sure. We'll make sure to send out slides. Okay, sure. Right. Okay, so to illustrate that this is important, there is something called leukocyte adhesion deficiency, and it results in mutations in the CD18 gene. CD18 is a subunit of an integrin that plays an important role in this binding of the leukocytes to the endothelium. So leukocyte adhesion deficiency, LAD, involves recurrent chronic bacterial infections and problems with wound healing. And the reason for that is that the neutrophils cannot move from the blood to the tissue. And Therefore, they cannot be in the tissues to fight these bacterial infections. These patients still have normal B and T cell responses because T cell antigen-presenting cell interactions can still occur in the absence of the neutrophils moving to the tissue. Okay. And then finally, let's talk about how do innate immune cells recognize pathogens. So innate immunity recognizes broad patterns. Innate immune cells act quickly, and they act non-specifically, which means they recognize broad categories of patterns as opposed to specific antigens the way the adaptive immune system does. So these patterns involve things like pathogen-associated molecular patterns, or PAMPs for short, damage-associated molecular patterns, or DAMPs. So PAMPs are things that result from the presence of pathogens, like viruses or bacteria. DAMPs are things that result from mechanical damage. So for example, you cut yourself, there's a bunch of dead cells, the dead cells spill out their insides, and that is DAMPs. And the major functions of recognizing these patterns is recognize a pathogen, destroy the pathogen. In contrast, adaptive immunity is extremely specific. It reacts specifically to a particular antigen. So with the innate immune system, you get recognition through pattern recognition receptors, or PRRs. These recognize broad classes of pathogens. They are expressed by all cells of a particular type. So this is an important difference between the innate immune system and the adaptive immune system. In the adaptive immune system, different B cells or T cells express receptors against different antigens. And very important thing about this is these innate receptors do not recognize self patterns. So they don't recognize anything that is found in the body outside of infection or damage. And then the important types of these receptors to know are the toll-like receptors, or TLRs. They detect bacteria and viruses. Nod-like receptors, or NLRs, recognize intracellular bacteria. And then C-type lectin receptors, CLRs, recognize fungus. And all these things feed into the master regulator of innate immunity, which is NF-kappa B. NF-kappa B, or nuclear factor kappa B, is a transcription factor that activates transcription of a multitude of pro-inflammatory genes. Okay. So toll-like receptors, or TLRs for short, recognize PAMPs, pathogen-associated molecular patterns. So these are things that pathogens have that our body doesn't have. So things like LPS, lipopolysaccharides, found on the surface of many bacteria, but not made by the human body. Non-methylated double-stranded DNA. Human and mammalian double-stranded DNA in general is methylated in a specific pattern. So when the body sees non-methylated double-stranded DNA, it has to have come from an intruding organism. Types of RNA and also flagellin. Flagellin is commonly found in bacteria because the motile kinds of bacteria propel themselves to flagella, and flagellin is a component of that. So the TLRs are expressed either in endosome inside the cell, as you can see here. These endosomal TLRs recognize foreign nucleic acids, foreign DNA and RNA, and then you have other TLRs that occur on the surface of the cell, and they recognize things like flagellin or LPS that would be found on the outside of the cell. So fun fact, toll-like receptors were actually first found in Drosophila, in fruit flies. And in fruit flies, the toll gene is actually necessary for specifying the dorsal ventral axis of the embryo. So when a Drosophila embryo is developing, it has to be able to tell which side is up and which side is down, and it uses toll signaling for that. And then much later, people discovered that this toll signaling from fruit flies is actually homologous to the toll signaling in humans and in mammals in general that fights infection. All right. So as I mentioned earlier, the toll-like receptors, upon activation, feed into this master regulator called NF-kappa B. So what happens is when the receptor is engaged by its ligand, it triggers this complex kinase cascade. A bunch of proteins get phosphorylated in sequence. And what happens is NF-kappa B, the transcription factor, in the absence of infection, it's sequestered in the cytoplasm by its sort of accessory inhibitory unit, I-kappa B. In the presence of signaling, I-kappa B is degraded and therefore releases NF-kappa B. And now NF-kappa B is free to go into the nucleus. And once it's in the nucleus, it can bind to its target genes and activate transcription. And it is the master transcription factor for a variety of pro-inflammatory genes. And then lastly, node-like receptors are intracellular sensors. So similar principle to the toll-like receptors, but they're found in the cytoplasm, and they specialize in detecting intracellular bacteria. They also feed into NF-kappa B signaling, but they can also activate cell death because, as you can imagine, if a cell is infected by intracellular bacteria, then it may be beneficial to the whole body to sacrifice that cell and trigger cell death rather than let the infection spread. All right, so thank you very much for your attention. Does anybody have any questions? Yes. Right, sure. So what is shown here is you've got a toll-like receptor that binds to a ligand. In this case, the ligand is LPS from a bacterium. This signaling attracts an adapter protein called MID88, and this adapter protein attracts further proteins, and you get a kinase cascade. The kinase cascade is basically one protein gets recruited, gets phosphorylated. That in turn enables it to phosphorylate the next protein downstream and so on. And once everything becomes phosphorylated in turn, it leads to the breakdown of I-kappa B. I-kappa B becomes targeted for proteasome-mediated degradation. And when I-kappa B is degraded, it releases its binding partner, NF-kappa B. NF-kappa B normally sits in the cytoplasm. But when I-kappa B is degraded, then NF-kappa B is free to move into the nucleus. Inside the nucleus, NF-kappa B acts as a transcription factor, and it turns on transcription of a bunch of genes that are involved in the inflammatory response, including cytokines and chemokines. So basically, the point of all this is cell detects a pathogen-associated molecule. Cell responds by turning on NF-kappa B and turning on a lot of different cytokines and chemokines that kick the immune response into there. Does that make sense? Yes. Okay. All right. Good. So thank you, everyone.